So today, our gene of interest is vascular endothelial growth factor. And the various functions of the vascular endothelial growth factor are that it is involved in angiogenesis, inducing vascular endothelial growth factor, facilitate development of capillaries, and contributes to facial growth, tissue repairs, wound healing. It also have anti-apoptotic effects, inhibit program cell death. Second function is it's associated with disease such as dysregulation linked to rheumatoid arthritis, etc. Uh, it has various isoform, that is A, B, C, D, and IGF. This slide is a schematic diagram explaining VEGF. We have taken it from Homo sapiens, means the humans, and this gene has a pivotal role in the angiogenesis and the mammalian expression vector with the CMV promoter. So we have used a pcDNA 3.1 plasmid. So this all is the 5428 or 5427 is the base pair size. Okay, so we have taken Chinese hamster ovary cell line. An introduction of is a common host for it's a common host for recombinant drug production, particularly monoclonal antibodies. It is also ability to perform post-translational modification. When the plasmid is designed for recombinant protein expression in CHO cells, typically contains a strong promoter and a selection marker ensure only plasmid containing cells are cultured. The novel approach used in this study is HR cloning method. Vector okay, so it is very this CHO cell is very important in biotechnology and pharmaceutical research, as it's versatile and adaptable to various cultures due to its post-translation modification, especially glycosylation. It also have advantage for correct folding and functionality of therapeutic proteins, and it also plays a role in diverse proteins such as an expression of diverse proteins such as antibodies, hormones, enzymes, etc. Okay, so cloning process, we use the pcDNA 3.1 high group, sorry, plasmid for subcloning of the heavy chain, and that is followed by digestion with the echo R5 restriction enzyme. The yellow marked area is a restriction site for the restriction enzyme. Then we use the primers of 1400 base pair. Yeah, but each primer consists of 40 nucleotides and 20 nucleotides that align with the plasmids, aligning with the plasmid sequence on both sides of the eqr5. And then primer sequence, the polymerase chain reaction for the heavy chain are we use the PCR implication for the heavy chain. Okay, so we use the universal colony PCR. The resultant product was subcloned here into pcDNA 3.1 neo through eqr1 and dimensional restriction enzyme. Here it is 1.1 agarose gel identified 1400 base pair product, and the 900 base pair product was cleaved. The pcDNA 3.1 high group was digested with eqr5 for 2 hours at 37 degrees C. The line 1 show the 5 digested plasmid, or the line 2 shows a 5 undigested plasmid. And here in the pcDNA 3.1 universal colony was performed to screen the recombinant colony. The lane 1 ladder and lane 2 show the PCR product was uploaded, and 1600 pm base pair band was seen right here. Okay, so in the last, the transfection of the recombinant plasmid, so we use the culture of CHO and 5 cell line in respective media. So distinct growth rates were observed, and so the K562 exhibit a faster growth than CHO cells. So the pcDNA 3.1, it new is for light chain, and the pcDNA 3.1 high growth is for heavy chain. So electroporation was used for introducing plasmid into K562 cell. So the CHO cell transfected using a mammalian transfection kit. So the transfected cells with neomycin and hydromycin antibiotics were used for the selection of the transfected cell. Okay, so in the last, So in the lab, the recombinant protein SDS page underwent concentrated sample were mixed with cold acetone centrifuge and then loading buffers. Lanes include 30 microliter of 24 hour, 48 hours, and K562 cells. While here the SDS, the lane one include the human IgG1. The lane two is empty. The M3 lane three recombinant antibody from CHO. The four include the 5 microliter protein marker, where the five include the recombinant antibody from K62. Here lane 1 30 microliter of 72 hour K562 culture medium, lane 2 5 microliter of protein, and lane 3 have 30 microliter of 72 hour CHO culture medium. Similarly here lane 1 have 5 microliter protein marker, lane 2 and 3 have 1 microgram of VEGF electrophoresis of 10% SDS page. So in the last, purification and Western blot analysis. So the culture media was subjected to purification through protein. So affinity chromatography was performed, and the antibody determination concentration was the K562 antibody was 41 microgram per milliliter, and the Chinese hamster ovary antibody was 35 microgram per milliliter. So a purified antibody that is 14 nanogram undergoes electrophoresis on a denaturing SDS polyacrylamide gel electrophoresis gel. So a transfer of protein into a so we transfer the protein to a nitrocellulose membrane. A Western blot analysis was conducted using the anti-human IgG1 HRP conjugated human body. Thank you. हाँ 10 मिनट तो ये बोलते हो गए ये उससे भी लंबी है